karakia timatanga. Tū tawa mai I runga, tū tawa mai I raro. Tū tawa mai I roto, tū tawa mai I waho. Kia tau ai, te mauri tū, te mauri ora, ki te katoa, haumi e hui e tae ki e. Come forth from below, within and from the environment, vitality and well-being for all, strengthened in unity. Ka tangi te titi, ka tangi te kāka, ka tangi hoki ahau, tihei, mauri ora. Ko te raroa me Ngāti whātua oku iwi, ko Ngāti Moroki me te uri o hau oku hapu, e noho ana au ki Rotorua. Ko Elise te huna ahau, ko kaimatanga mātai taiao mo te papa atawhai. Nau mai hara mai, koutou, I tēnei pō mo te kaupapa, Tūhuratia te taiao, Tūrua te Aotearoa me pūtaiao I te hihi. Tēnā koutou katoa, my name is Elise Te Huna and I would like to welcome you all to the second Hihi Conservation Science Event, which is celebrating one of the themes of this year's Conservation Week, Tūhuratia Te Taiao, Take a Moment to Discover Nature. I am a technical advisor for Te Papa Atawhai Department of Conservation and I'm based in Rotorua in New Zealand. I am the Recovery Group's Framework Coordinator and provide support to 18 threatened species recovery groups that are based all around Aotearoa, New Zealand. Department of Conservation DOC has a small but arguably important role in the Hihi Recovery Group. Recovery groups have been used in Aotearoa, New Zealand since the 1980s, initially to drive DOC work, but in the last few decades, they are increasingly used to coordinate a range of groups who are managing species. The Hihi Recovery Group and the associated Hihi Conservation Charitable Trust are passionate about sharing their knowledge and appreciation of Hihi with everyone, and this event is one way they have thought to do this. This event will focus on the latest Hihi research work that they encourage and support. The Zoological Society of ZSL is also a substantial partner here. On the surface, this might seem surprising given ZSL is on the other side of the world from Aotearoa, New Zealand, but it has strong links to Aotearoa, New Zealand through John Ewan, who is from Aotearoa, New Zealand and has a passion for Hihi. And over time, ZSL has hosted many researchers who have worked on Hihi. We will hear from some of those researchers tonight. Hihi was once much more common, found throughout northern Aotearoa, New Zealand. However, as with so many other species, it has suffered following European colonization and has become restricted to a single small population on the offshore island of Te Hauturu o Toi, Little Barrier. Since the 1980s, the government and then increasingly community conservation groups have been helping recover the species via reintroduction and providing intensive support to these new populations. Hihi are unusually challenging compared to most Aotearoa New Zealand birds, but we are making progress. There are now 8 populations, 7 of which are reintroduced and which all require ongoing support. These reintroduction populations combined have now increased the adult population size by more than 600 birds. They are well spaced across northern New Zealand and therefore much more possible to enjoy. So I recommend if you haven't seen a hihi before, if you're visiting New Zealand or you're in New Zealand or you're from New Zealand, and as our lockdown relaxes, that you take the opportunity to go and find one. You won't be disappointed. I will now hand over to John, Faye and Ashley as our hosts. Kia ora.
Kia ora, everyone. Thank you very much for joining. I think our panelists can turn on their videos now so we can see who we've all got here to hear from. Elise, thank you very much for that wonderful welcome and the brief background to species recovery projects in Aotearoa, New Zealand, the Hihi Recovery Program in particular, and how our Hihi family works together for the species. And so, although tonight is all about our fascinating, exciting current Hihi research, Elise has highlighted something that I just want to spend a small moment focusing on. Now, there are so many people that are involved with Hihi management. ranging from the New Zealand government through its Department of Conservation, our Hihi Conservation Charitable Trust, and importantly, a number of absolutely dedicated community conservation projects, which are spread between Wellington and Auckland, and they care for a growing number of reintroduced hihi populations. So before I go any further, I just want to say thank you to Elise for the support that she provides via the Department of Conservation. And thank you to everybody who actually dedicates their effort to ensure that hihi have a bright future. And so I'm really excited to be co-hosting this event this morning or tonight, wherever you are in the world, alongside Faye and Ashley, who are both here on the screen. Both Faye and Ashley are current PhD students that work with the hihi. And we have a super lineup of current hihi researchers, which are all these other faces on the screen. And we should really get cracking and hear from them, not from me. One other thing I just need to add, in case you are new to Zoom, or you need a reminder, as an attendee, as the audience, there are two important ways that you can communicate with us. And if you look down the bottom of your screen, there are at least two tabs. One is a chat and one is a Q&A or question and answer. If you can open those and please interact with us using them, that would be fantastic. Use the chat to say hello, to chat amongst everybody, to share information, share ideas. But please use the Q&A to send questions for our speakers, and Faye, Ashley, and myself will try and ensure that those are all answered throughout this event. So that's it for me for now. I will hand over to Ashley, who is in charge of introducing our speakers tonight. I hope you all enjoy. Thank you.
Thanks, John, and thanks, Elise. And so our first up is Caitlin Andrews, who's from the University of Cambridge and Zoological Society of London. And she's kicking off the research talks of her presentation titled "Who Heals the Forest? Individual hihi Differ in Pollination Behaviours." And Caitlin is currently based in the US, so hats off to her for joining us in the middle of the night. And just take it away as soon as you're ready, Caitlin. Thanks.
Thanks, Ashley. All right, so as Ashley mentioned, I'll be sharing some of the work I did studying hihi's role in pollination during my PhD, working with John Ewan and Rose Thorogood. The New Zealand of today looks very little like the New Zealand where hihi once flourished. The landscape has been heavily modified by human activities, leaving only small patches of remaining forest where hihi and other native birds can survive. So when we translocate hihi, we look for these patches of paradise where they have their best chance of thriving and hopefully going on to produce lots of these adorable chicks. But hihi translocations have another goal that we don't talk about as often. Hihi are important pollinators of native plants. So when they and other native birds were first lost from these ecosystems, it had devastating consequences for ecosystem health. So by returning hihi to these ecosystems, we're hoping that hihi will help to heal the forests for the benefit of all species. However, one large question remains. Is every hihi translocation equally successful at healing the forest? So when we translocate hihi, we're only bringing small numbers of individuals with us. And there's growing evidence that individual animals can vary in their contributions to ecological function, including pollination. So in our case, if some hihi are better pollinators than others, and we translocate these birds to start new populations, we may have an even greater chance of helping hihi to heal the forest. But how do we figure out who these special forest healers are? So in a previous study, I conducted a pollination experiment at one of our largest hihi populations. And what I did was I compared pollination outcomes across hihi territories. So we selected 50 hihi territories. And what we found was that some territories had better pollination outcomes than others. And since hihi chase intruders off their territories during the breeding season, we could assume that most of the pollination on the territory was due to the resident male and female on that territory. So the fact that pollination varies across these territories suggests that individual hihi might actually vary in their quality as pollinators. So in the present study, we returned to these same 50 territories to see if we could identify superior pollinators based on their behaviors. On each territory, we placed a pentagon of 5 artificial flowers, with only one providing a sugar water reward. And each time a hihi visited, we looked to see how they searched for food. So some birds used a systematic search strategy, for example, by always looking for food and searching the flowers in a clockwise direction. So from a plant's perspective, this could be beneficial to pollination because the pollinator is spreading pollen far and wide without returning back to the same plant within a short time period. Whereas a pollinator that searches randomly is more likely to make an error by going back to a flower that they've just emptied or found to be empty. So across all territories, we looked for patterns in how birds searched based on their age and sex, with adult males, adult females, and first-year birds all participating. So what did we find? Well, first, we had predicted that birds would vary in their search strategies, but for the most part, hihi tend to use a systematic search. So this is pretty impressive and maybe why hihi are such good pollinators to begin with. But nevertheless, we found that individuals did vary in their tendency to make errors by returning to flowers that they should have already known were emptied, so flowers that they had already visited recently. Adult males had a pretty low error rate compared to adult females and first-year birds. We think that this might be because females and young birds are subordinate, so they might have to divide their attention between foraging and keeping watch for predators and competitors, which pose less of a threat to males. So when they divide their attention, they may be more likely to make an error and lose track of which flowers they've visited. But these same individuals were actually better in another aspect of the task. They were more persistent. So males were more likely to give up before they actually found the food, while females and first-year birds usually persisted until they found the reward. And again, because they're subordinate, they may have fewer options of where to forage, so they may be more motivated to make the most of each foraging patch. So how do these behaviors actually translate into pollination? So in our previous study, we found that territories vary in their pollination outcomes. And here we found that individual hihi vary in the strategies they use while foraging. But while we predicted that good territories would be home to strategic foragers, we didn't actually find this relationship. Sometimes hihi on good territories perform very poorly in our task, and vice versa. So to return to the opening question of who heals the forest, we know from past work that hihi are forest healers by nature. But the present study suggests that individuals may vary in their contributions to pollination. So while we have not yet determined which behaviors make some hihi better pollinators, the fact that there is variation means that not every hihi translocation will necessarily be equally effective at restoring the forest. It will depend on which individuals we translocate. So over time, if we can work to identify what behaviors make hihi good pollinators, it may help us to help them heal the forest even more effectively if we can promote these behaviors through our conservation efforts. So just to wrap up, I'd like to say a quick thank you to the following people for their help in the field, and all of you for joining us to celebrate this awesome endemic bird. Thanks.
Thank you, Caitlin. That was great. I love those animations and illustrations. It worked really well. So if anybody has any questions, please stick them in the Q&A. Oh, we've got some already coming through. So from an anonymous attendee, a mystery questioner, were you able to track the juveniles to the point where you could say if they were male or female and how their foraging differed with age?
Yeah, so here we were mostly focused on birds that had already settled onto territories. So they were at the point where we could identify the sex. But yeah, that would be a great idea for the future, is to actually track how the hihi's role in pollination develops over the course of their lives, because juveniles definitely have different behaviors, and maybe they get better at pollination over time, or maybe there's something special about juveniles that we don't know might make them really good pollinators. So yeah, that's a great question.
It's cool. Yeah, you'd imagine that they might get better as they learn. So we've got a question from Priscilla. Is there any data on how other pollinators might help or hinder the pollination success of hihi? So other pollinating birds, perhaps insects?
Yeah, so I think, so in the study that I mentioned from previously, I was looking at quantifying hihi's role in pollination, and it seems that other birds can fill in some of the gaps that are left behind when hihi are lost from the ecosystem, but they don't necessarily do as good of a job at promoting pollination quality. So hihi seem to bring something special to the ecosystem by helping plants to produce extra high quality seeds. So that other birds might not be hindering hihi, but they just might not be doing as good of a job as the hihi do.
So I think we have time for one more question. So a question from Scott. Speaking about other pollinators, did you control for the other pollinators that were in the area, such as Tui and Korimako?
Yep. So again, by doing it on hihi territories, we were hoping to kind of isolate the effects of hihi as much as possible because they do chase intruders off the territories. But yeah, I used, we kind of developed a system to kind of control for other pollinators. So we quantified the contribution of insects to pollination by excluding birds from certain flowers, and then left other flowers open to birds and insects, and then compared pollination across those to kind of isolate the effects of birds. And we did find that almost all visitation by birds to these plants that we were focused on was by hihi. So we can kind of be more confident that it is due to them.
Yeah, that's a really neat experiment. So sorry, we're going to have to leave some of these questions to the panel at the end. So we're on a bit of a time limit. So we'll move on to Ashley for the next speaker.
Yeah, so thanks for that, Caitlin. That was great. And thanks for answering those questions. And so our next speaker is Elizabeth Parlato. So she spoke last year as well, if anyone was here and remembers her talk. And she is a postdoctoral fellow from Massey University. And we'll be speaking today about whether individual variation is relevant to hihi population dynamics. So whenever you're ready, Liz, just work away.
Great, thank you. Right, well, kia ora. My name is Liz Parlato, and I'm going to talk to you about some work that Doug Armstrong, Peter Frost, and I have done looking at whether individual variation is important in terms of hihi population dynamics. So I'll firstly just explain what individual variation is. So individual variation relates to differences among individuals in survival and reproduction rates that are not related to the individual's age or its sex. So then the question is, why might we think that individual variation could be important for hihi population dynamics? Well, we know that in some hihi populations, survival, detection, and reproduction rates can vary quite a lot among different individuals. And given this, if we ignore it, we could end up with biased estimates of age effects and or population size. So we decided to investigate the importance of individual variation on hihi population dynamics by focusing on the Bushy Park hihi population. So just to give you a bit of history about this population, 44 hihi were released into Bushy Park in 2013, and there was a reinforcement of 10 birds in 2018. So from the intensive monitoring that's taken place over the past 8 years, it's clear that there are really some quite big differences among individuals in terms of their reproduction success, their longevity, and their detection probabilities. So we thought this would be a good population to investigate whether individual variation is important for the population's overall dynamics. So to do this, we used two alternative approaches. The first was developing a population model that didn't include individual variation. This model did include sex and age effects, which meant that the sex of an individual and its age were taken into account when estimating survival, detection, and reproduction. The second approach was to develop a model that included individual variation. That is, that it estimated effects for each individual in the population. And in addition to individual variation, this model also included sex and age effects, like in the first model. So I'll just talk you through now what we found. So these graphs here come from the first model, which didn't include individual variation. And if you look at the top graph, that's showing you the mean number of fledglings per female per year. And you can see that older females were estimated to produce a lot more fledglings than first-year females. So the older females there are the ones in the darker pink curve. Then if you look at the next graph down, you can see that males, which are shown by the blue curve, they have a higher survival probability than females, and that fledglings, shown in the grey curve, they have the lowest estimated survival probability. And then going down to the bottom graph, this is showing detection probability. So that's essentially the probability of detecting a bird in a survey if it's present in the population. And you can see here that males were estimated to have a higher detection probability than females. So let's compare this with our other model where we did include individual variation. So you can see, looking at the top graph, the distributions have changed really quite substantially. And the age effect has actually disappeared altogether. And so essentially what you can see there is that for an average female, the number of fledglings produced was much lower than under the other model. We move down to the survival model, the mean male survival probability remains the highest, but the age effect that we previously found has disappeared, because you can see there that adult females and fledglings were estimated to have very similar survival probabilities. And moving down to detection probability, here the sex effect has disappeared altogether, with females and males essentially having the same detection probabilities. And you can see that that curve is really flattened out quite a lot, and that essentially is showing that some birds are really quite conspicuous and really easy to find regularly, and some other birds are really quite elusive. So what does it mean in terms of the overall population dynamics? So this graph here shows you the number of females estimated to be in the population each year from 2013 right through projected until 2023. So the solid lines that you see there are the years for which we had data available, and the dotted lines are projected population sizes into the future. And the pink lines, the pink line is the one with individual variation, and black is without. So you can see from the solid lines that really the two models provided very similar estimates for the years for which we had data. And then with the projections, the model with individual variation produced slightly more optimistic results than the model without. So just to conclude what all this showed us, we found that there was really clear evidence of individual variation in the bushy park population, and that there was a relatively small portion of those individuals which were high quality. And I just wanted to draw special attention to this very special female here. So her band combination is BMW, and she was released in 2013, one of the first birds released. And she has been a brilliant survivor. She survived right through, and she was detected in the most recent survey. And she's also been an incredibly good producer of fledglings, producing between 5 and 12 fledglings each breeding season. So she definitely qualifies as a high quality individual in the population. She's pretty amazing. And what we found was that accounting for this individual variation really did change our understanding of the population's demographics in terms of the survival and reproduction rates, that our understanding of what was happening with those. But overall, it didn't have a huge impact on the overall predictions of the population's dynamics. And that's really quite reassuring, because it means that a model with or without individual variation is going to give us a good handle of what's happening with the population, and will be a good basis on which to make management decisions. So I'll just finish off by saying thank you so much for being part of this, and thanks so much for listening.
Thank you, Liz, for that wonderful talk. Everybody stick their questions in the Q&A. We have one from Ruth that asks how long the study was that this data is coming from.
So we had, I think it was about 8 years worth of data that we were working off. Yeah, which is quite a decent.
So yeah, plenty to look at age effects.
Yeah, which is brilliant. Yeah.
Yeah, that's great.
Amazing monitoring that goes on there. So yeah, all the females are tracked through the breeding season, and we do 2 to 3 surveys every year as well to try and keep track of the birds present in the population. So yeah, no, a lot of work goes into it.
Yeah, I have a question as well. So these high quality individuals that you talked about, what do you think that it is about them? Do you think there could be like genetic signatures of that, or do you think it's something that happened in their fledgling phase or nestling phase, like the quality of their parents, or do you have any ideas about that?
We haven't really looked into that yet. So not sure. I mean, it could just be like a chance, you know, you just get these amazingly good birds that come through, and like they are quite remarkable, and they have a huge impact on the population's dynamics. Like, you know, they really, there was quite a close correlation between good survivors and high producers. So like it was really interesting to see how you do get this quality coming through. As for what underlies it, yeah, I'm not sure. I mean, it'd be really interesting to look into whether it's heritable. So whether offspring of those high quality individuals also tend to be have positive effects in terms of their survival and reproduction as well. So yeah, that'd be really interesting next steps to take this forward.
Cool, yeah. We've got a few more questions. A question from Rod. Any thought of modelling loss of genetic variation over time, given the big asymmetry in reproductive success? So kind of like what we were talking about, those super productive individuals, are they kind of swamping the population with their genes? I think that's what.
Yeah, absolutely. And that could very much be the case. And I think, in fact, some work's been done on Tūhuratia, which I shouldn't quote too much, but I'm aware that a female that was brought in from Hauturu to sort of bring in a bit of extra genetic variation, she actually, I think, turned out to be an incredibly high quality female and produced a huge number of fledglings, and so a lot of her offspring are now in the population. So yeah, it's amazing how that can have an influence on your genetic diversity in your population when you just do have a small selection of females which are overrepresented in terms of their reproduction. Yeah.
And we're going to have to leave the questions there. Very sorry.
Ashley has spoken.
I know I'm being the nasty timekeeper this evening, but there is time at the end with our panel discussion to answer some more, and also they can be answered in the Q&A as well. But yeah, I'm going to have to be strict, I'm afraid. And but thank you so much, Liz. That was amazing. And
thank you. Thanks for the great questions.
Yeah, there was so many questions. Thanks again to our audience for that. But our next speaker up is Vix Franks, who's from the Zoological Society of London. And she also put together some of the lovely Hihi artwork that you'd have seen in all our posters and stuff. So big thanks to her for that. And her talk has the slightly sci-fi sounding title of Human versus Machine. So looking forward to that. And take it away, Vix, as soon as you're ready. Thank you.
Thanks very much, Ashley. I'll just get my screen shared. Perfect. Right. Hi, everyone. Thanks very much for listening in. Before I start, I'd just like to say a really big thank you to the groups that have made this work possible, particularly the team at Shakespeare, but also the Hihi Conservation Charitable Trust and Auckland Council. And it's because of you that I can do this research. So when we move or translocate animals from one site to another to establish new populations, our responsibility doesn't end when we release the animals into their new home. Post-release monitoring is essential if we want to know how our population is doing, because we might lose animals if the population unfortunately declines, but equally the animals might establish and thrive. And in either case, we need to know these outcomes because it can help us plan and improve future translocations. And so this information from post-release monitoring can be really important. Now, there are a variety of ways that we can complete this monitoring goal. And one option is using surveys where a person physically goes out and attempts to find all the alive individuals in the population. But increasingly, technology is being used as a way to assist by remotely monitoring animals, for example, using camera traps or tracking tags. Now, each of these methods has their own pros and cons, and evaluating these can help us choose the most efficient monitoring approach to get the best information for future. And we can think about this question in a variety of different ways, but today I'm just going to show you one area that we've been considering. The translocation I'm using as an example happened last year, actually, when 40 juvenile hihi were moved from Tūhuratia Mātangi Island to set up a new population at Shakespeare Regional Park. And I'd like to introduce you to our two monitoring contenders for this human versus machines comparison. So firstly, to date, post-translocation Hihi surveys have been carried out by a person on foot. And in recent years, this has been done by our awesome Hihi Conservation Officer, Barry McCready. However, for this translocation, we also introduced an alternative monitoring approach for the first time. And all hihi were given a little microchip RFID tag in a leg band, which equips each bird with its own barcode effectively. And this allows us to log each visit made by every individual to the 3 supplementary feeders that were in the park. So following the release of hihi into their new home, VARI carried out 4 surveys on foot up to September 2020. And at the same time, the RFID loggers were recording at the feeders and logging which birds were visiting. And they've actually been doing this continuously since the release happened. So what I'm going to show you now is a quick comparison of the numbers of hihi recorded by each of the monitoring methods to show you how they help us track how the population was doing following the release. So this first graph is going to show the numbers of individual hihi recorded at the feeders each day using the RFID system, and that's over the entire first year post-release. So what you can see from this is that we get a really detailed continuous tracking of the number of birds that are using the feeder across this period of time. However, how do we know if this is representative of what the real population is actually doing? Well, if we add in the in-person surveys as red crosses, we can then compare the numbers. And what we can see is that the numbers recorded by the RFID mostly reflects what was going on during these intensive bursts of on-foot in-person surveys. However, if you notice, I said mostly, and I'd just like to highlight the first survey where something quite different happened. And we can see that there's a big discrepancy between the number of hihi recorded by the human surveys, which is the red cross at the top, which recorded 39 birds. And this is in comparison to the RFID, which only recorded 1 bird in this period. So this indicates that the hihi were potentially behaving differently at this point, and they didn't seem to be using the feeders and weren't picked up by the RFID loggers, but they were still spotted in the park on the wider human surveys. So this first comparison has therefore highlighted some good things about using the RFID system, in that when the birds were using the feeders, RFID provided a reliable gauge of how the population was doing on a daily basis. But there's also some negatives, and that we definitely need the birds to be using the feeders in order for this technology to be able to work. So to summarize what I've talked about here then, when we use different monitoring approaches might be important, and this also might depend very much on the behavior of the animals that we're trying to monitor, which can also vary immediately after a release following translocation and then into the longer term. However, in this case, once hihi had locked onto the feeders and the whole population was using them, it provided a good way to track every individual. And this might highlight a main advantage of this RFID system, in that this system can be left out for a much longer period of time and record continuously, and then we can look at how populations track over this period. I still have much more to look at, as I highlighted at the beginning. There's a lot of different ways that we can take these comparisons. And so basically this human versus machine is continuing. And I'd just like to say thank you very much for listening.
Oh, I did a John. Thank you, Vix.
No problem.
That was great. Sorry, John. So we already have, thank you. Just got a cup of tea delivered.
Important to keep going, Hansi.
Sorry. So we've got a question from Caleb. I hope I'm pronouncing that properly. Is the decline of hihi due to death, or is it just because the hihi are stopping using the feeding sites?
Yeah, that's a really good point. And luckily having these 2 different monitoring approaches that we're using helped us to kind of ground truth what the RFID tags were showing. And from the line from how the human monitoring compares to the RFID, it looks like we are, unfortunately, we have been losing those individuals. There are also, we know from the breeding population, the breeding sort of time, that there were a few individuals there, which can also sort of add a little bit of a ground truthing of that later on.
Yeah. So many, this is the most engaged audience I've ever seen. So many questions
great, isn't
coming
it?
in.
It's
It's really
really good.
great. Sarah Jane asks, how are the feeders spread out, and could their territorial behaviour affect which individuals are using them?
Yeah, definitely. And this might be clearer in a more established population. So for example, on Tiri, we definitely know that there are individuals there which prefer certain feeders, and you're to the point that you'll always see those individuals at particular feeders. So it's likely that that's also happening in other sites like Shakespeare. And it's actually something that we're going to be looking into hopefully a bit more as we go forwards, because we do have all of the individual information on these birds. We can see if there's preferences in what site they're using.
Yeah, that would be really interesting. We'll squeeze one more in. So this is from an anonymous person. Do you see any drop in the RFID readings when there are periods of high nectar availability?
Yeah, that's a really good question as well, actually. And I don't know if it was very clear on that graph that I plotted with just the sort of numbers of birds over time, but there does seem to be these peaks and troughs in it. And it's definitely interesting to think about whether that might be that the birds aren't using the feeders as much. I mean, we know again from places like Tiri that when there is lots of natural forage out, the feeder use does drop off. So you'd expect to see it here and hopefully get that reflected in the RFID readings.
Mm-hmm. Yeah. Really interesting questions. We'll have to come back to these at the panel discussion. So I'll hand over to Ashley now.
Thank you.
Yeah, that's great. I feel bad for interrupting the questions, but yeah, keep sending them in and we'll get to them hopefully in the event. But next up, we have a first-time speaker at this event. So it's really great to hear about her research. So Sarah Bailey is a master's student at the University of Auckland, and her talk is titled New Foundations for Hihi Conservation Genomics, How We Built High-Quality Reference Genomes and How They Will Help. And that sounds really fascinating, Sarah. So just go ahead as soon as you're ready.
Cool, kia ora koutou. Today I'm going to be talking about new foundations for Hihi Conservation Genomics, how we built the high quality reference genomes and how they will help. So first off, what is a genome? So the characteristics of all living organisms are determined by their genetic material and their interactions with the environment. And a genome is the complete set of this genetic material in an organism. A reference genome, is a genome that has been sequenced and assembled so that we have a digital copy available. And when we study a species using whole genome information, that is genomics. To build a reference genome, there are two main stages, and this is sequencing and assembly. For sequencing, we took blood samples from a male and a female hihi. We extracted DNA from those blood samples and then loaded this DNA onto a sequencing device, which records the bases in the DNA and uploads it to a computer. We can't sequence whole chromosomes at the moment. We can only sequence fragments, and these are called reads. So once we have our reads loaded onto a computer, we can start the assembly stage. And this stage uses computational steps to arrange the reads into larger pieces of DNA, known as contigs. And this stands for contiguous sequence with no gaps. And that is what our genome is made up of. The quality of a genome is typically assessed using contiguity and completeness. Contiguity looks at the number of pieces in the genome. And looking down here at these three karyotypes of these duck species, this here is a chromosome, and these two similar looking chromosomes are a chromosome pair. And in birds, there are 40 of these. Birds also have ZW sex determination, so males have 2 Z chromosomes, and females have 1 Z and 1 W chromosome. So these three karyotypes are of females. In an assembly, each chromosome pair is combined into a consensus chromosome, and each contig represents a piece of this consensus chromosome. And it is better if an assembly is made up of the smallest number of pieces possible. For completeness, which evaluates how much of the genome is represented by the pieces of the assembly, we can evaluate this by searching the genome for a set of core genes that are expected to be found in all bird species. And for a genome to be complete, we want to find over 90% of these genes. So we built 2 assemblies, one for a male hihi and one for a female hihi. And the male genome size of around 1.05 billion base pairs is slightly smaller than the female genome size of around 1.06 billion base pairs, which is due to the presence of the extra W chromosome in her genome. Looking at the number of pieces that the genomes are assembled into, the male has 617 contigs, which is a little bit higher than the female genome with 522 contigs. Then looking at gene completeness, we reach over 90%, which is great. And the male genome has a gene completeness of 96.7%, which is slightly higher than the female genome, which has 95.6% gene completeness. And we can use these reference genomes for conservation genomics. We can anchor whole genome resequencing data from other individuals to the genomes, and we can study genetic diversity using this data. We could look at the diversity of immunity genes, and if we specify what genes are lacking diversity, because it's theorized that a species is better off if it has higher genetic diversity, we can try and identify what threats hihi might be vulnerable to in terms of disease susceptibility. I'd like to acknowledge Ngāti Manohiri as kaitiaki of hihi and thank them for their support. I'd also like to thank Nessie, Genomics Aotearoa, my 2 amazing supervisors, and the organizers of this event today.
Thank you, Sarah. That was great. And yeah, this work is really useful. I've just used this in my lab work as well. So it's really valuable stuff. So a question from an anonymous attendee. The reference male and female that you used, were they taken from Little Barrier Island?
Yes, they're taken from Te Hauturu o Toi, and I think they were sampled in 2018. I could be wrong there though, yeah. Mm-hmm.
So you might expect differences between those birds and birds that you find in the reintroduced populations?
Yeah, I would say so. There's definitely individual variation, and that's kind of the benefit of taking the resequencing data and aligning it to the genome, because then you can kind of grasp more of what the whole population looks like rather than just these 2 reference individuals that will look slightly different.
Yeah, and I've already said that I found the work that you're doing really useful with my work, looking at age effects. Are there any other kind of goals that you have for this, this like genome sequence to be used? Or
I know Laura's using it for her inbreeding analyses, and I guess since the reference genome is kind of like a catalog of the coding and non-coding information in Hihi, you can use it to look at all sorts of questions. So not just conservation questions, but you could look at more evolutionary questions, like what are the genetic bases of some of the really interesting traits that Hihi has, like I guess coloration, or maybe some of the interesting like or other behaviors like nectar feeding and things like that they have.
Yeah, that would be really interesting. We'll take one more question from Sahar. Have you seen or expected subspecies within the New Zealand populations of hihi?
We haven't looked at that, and I wouldn't know. Yeah, we'd have to, I guess, get information from other individuals. Yeah, I'm not really the person who would be able to answer that, sorry.
Well, maybe we can come back to that in the panel discussion and
yeah,
can chip in with their expertise. All right, let's hand over to Ashley to introduce our final speaker.
Thanks very much, Faye, and thanks, Sarah. That was really interesting. And yeah, it's great that the work is going to be used for lots of other people, which is the goal. So our final speaker, or our final talk for our panel, is by Laura Dunch, who is a PhD candidate who's based along Sarah at the University of Auckland. And her talk is titled From Pedigree to Genomics, Advances in Measuring Hihi Inbreeding. And that sounds like a great way to finish off the research talks before the panel. So work away, Laura, as soon as you're ready. Thanks.
Cool. Can you see the screen?
Yes.
Just making sure. Hi, everyone. Yes, my name is Laura. For those of you who don't know me, I'm a PhD student at the University of Auckland. I'm originally from Germany and came here to New Zealand to study Hihi genetics, which means that I'm not a field scientist, but sitting in front of the computer most of the day using bioinformatics tools. And my talk today will be about how we measure inbreeding in Hihi. Let's first disseminate some of the words in my talk title to make sure that we're all on the same page. So when I refer to a pedigree, what I mean is a record of family relationships. So think 23andMe, but for birds. The second word you might hear me say a lot is genomics. I believe Sarah has just defined it for you quite beautifully. But in short, it means that someone is studying all the DNA of an individual. And lastly, there's the word inbreeding, which refers to the mating of two individuals that are closely related. So for example, cousins or even brother and sister Hihi. And inbreeding is revealed by low variation in the genome of an individual. So threatened species usually exist only in small populations and hence are potentially facing inbreeding, which in those small populations is directly related to overall genetic diversity. And as we all know, genetic diversity is important and something conservation scientists and managers are trying to preserve. Now you may ask, how does inbreeding look like on paper? While traditionally researchers would go out and observe birds and construct a pedigree and then estimate relatedness and inbreeding from there, nowadays the use of genomics allows us to improve our inbreeding measures in 3 different ways. So firstly, genomics give more accurate estimates of inbreeding, as it doesn't have a starting ancestor like an observed pedigree would have. And secondly, they give inbreeding values for individuals we don't have any pedigree information for or might have only missing information. And thirdly, Hihi have quite a high rate of extra pair paternity, so meaning the bird that takes care of the chicks might not be the actual father, and genomics can tell us about that too. So let me walk you through an example of an inbreeding event here. So in generation 1, we have mom and dad Hihi, and then they produce offspring, and each parent passes on a random half of their genetic material. And then you have generation 3, where the offspring, which is the offspring of those 2 siblings, a male Hihi, there you go, that has by chance inherited 2 chromosomes from the same ancestor. So we now consider this Hihi inbred. And here's how we measure inbreeding and report inbreeding by using something that we call the inbreeding coefficient F. So F can range from 0 to 1, where 0 means that a bird has no inbreeding history and values higher than usually around 0.5, describe quite high inbreeding levels. So in this example, this female has a value of F of 0.29, so it's slightly inbred. So the goal for one of my PhD chapters is to measure inbreeding in 5 Hihi populations with individuals taken from the last not quite 20 years. And basically you want to see how often the scenario I just described really happened in reality. So I used genomic data to infer individual levels of inbreeding for Hihi from 5 populations, which you can see here as dots on the map. And this plot shows the inbreeding value F on the y-axis, and the black line in those boxes shows the average inbreeding value for that specific population. And what you can see here is that birds in the remnant population, so on Tūhuratia, the black box, they are the least inbred, and birds in Tūhuratia Matangi have the widest range of inbreeding values. Overall, we find that small, older translocated Hihi populations, such as the one in Zealandia, which is here noted as Karori, and on Kapiti Island have higher inbreeding levels than the much larger population on Tūhuratia and the more recently established population in the Mangotautari sanctuary. So in future projects, I want to see whether less inbred birds are doing better with regards to offspring numbers and survival compared to Hihi with higher inbreeding levels. So of course, the Hihi is not the only species of conservation concern where inbreeding is measured. As you can see here, there are other threatened species that are doing a little better and some are doing much worse. This has mostly to do with their population numbers, but also can be quite affected by analysis parameters, etc. So we tend to not compare inbreeding values directly between species, as they all have very different population histories. Thank you all for your attention, and I'm happy to take any questions.
Thank you, Laura. You did a great job of explaining some really complicated stuff really simply there. So we'll move straight on to questions. So from an anonymous attendee, doesn't this study actually show a bottleneck effect rather than inbreeding specifically? So maybe you can talk about those two things and what they mean.
I'm not too sure what.
So I think
referring to.
Well, a bottleneck will result in inbreeding. So I think this person is.
So yes, so inbreeding can be a consequence of bottleneck effects. So it usually only occurs in small populations. So if you imagine you have a large population and you're translocating birds to a new place, chances are that if that population is relatively small, that you are more likely to breed with someone that, yeah, is somehow related to you. So inbreeding is a consequence of it. So it's always sort of related with each other most of the time. Yeah, but it's not necessarily the same thing.
Yeah.
I'm not sure if that answered the question. But
different facts, but hard to separate them because they're kind of linked. Yes. So we'll move on to a question from Eric. Is there regular periodic moving of individuals between populations to introduce new genetics?
No, so he actually don't migrate, and they don't definitely don't migrate between those different populations. They're also always quite far away from each other, so they stick to the fenced sanctuary that they're in, but there's no natural migration between the populations, which is in a way cool. So when you do these population studies, you know that the birds stay in that specific population and they don't actually come from a different one than what you thought they do.
Mm-hmm. Yeah, and kind of linked to that is a question from Megan. Do we need to start mixing birds like manually between the populations? And would that help, I think, is that a question?
I guess still haven't assessed how their inbreeding is actually affecting the bird's fitness. So sometimes inbreeding might occur, but it doesn't actually have a big effect on the bird's fitness. Sometimes individuals that are actually very inbred are what we call purged from the population, so they don't actually reproduce and don't really pass on bad genes onto the next generation. It is not currently something that's included in translocation plans, the genetics side of things, but it is definitely in other populations or in other species a measure that's sometimes undertaken in order to increase genetic diversity.
Mm-hmm. Yeah, so we don't know yet whether it would help them or not. Still need to look into that.
Exactly.
We have so many questions, but we'll have to move on to the panel discussion now, which is unfortunately going to be really short because we've just, it's had so many.
Yeah, thank you very much. Thank you very much, Ashley and Faye, for coordinating and keeping us more or less to time. And thank you very much for all of the awesome presentations. I think they ran smoothly. They were fantastic, really good animations, really good slides, and very varied in topic. And I hope that came across. The research work that's done to support Hihi recovery is very varied. We've been managing Hihi and had a heavy research focus for decades now. And I think there's a comment in the chat around there's always so much to learn. And that's very true. So the more we're just scratching the surface, which is very good for Hihi. We keep improving what we're doing for management. And it's also very good as a showcase for other species recovery programs and what they might be able to learn from us. as much as we learn from them. We really are down on time, so we've got a couple of minutes before I'm handing over to Elise to close the meeting for us. So I'm not sure how best to do it. I don't think in the end we can have a proper panel discussion, but there are a couple of things that were raised more generally. There were questions around where some places are. I know Bushy Park has now been answered in the quick Q&A, but please do go and have a look at hihiconservation.com website, download that report that I provided a link to in the chat at the beginning. It provides a map of all of the current reintroduced populations and information about those sites and how you can go there, how you can visit, see the birds. They are fantastic. And also how you might be able to get involved if you would like in this Hihi family and helping recover this bird. Hopefully what's come across strongly, one message that I'd have is how we try and use the research work we do to support the recovery of species. And I think that shone through in the presentations tonight. It's certainly something that as co-chair of the recovery group, we find critically important, where the success we have is in part because of all of this hard-won research that's been done by all these cool people. And we always like to challenge the researchers to make sure that their research is meaningful for Hihi recovery, as well as just having a neat study system to work with. Lots of effort goes into monitoring. We need to make sure our monitoring is useful for management. And that's another key message I hope can come across tonight. And it's something that I'm particularly passionate about. Controversial point, there was a neat point raised in the chat about getting these birds down to the South Island. One of my side interests is around the role of assisted colonisation and species recovery. There has been previous research looking at future suitable habitats for this little bird. That does include the South Island. We've got no current plans to do anything about that, but it is sitting there as an idea. Laura touched on genetic management. We are right now trying to set up the next 20 years of Hihi recovery, and that certainly does have a lens on genetic management as well, and some ideas around shuffling and mixing up the birds between populations. That has been and will continue to be informed by the cool work that people like Laura and Sarah and many others are contributing. Honestly, we're 1 minute to go, so I am unfortunately going to have to stop talking, stop all this cool conversation and hand over to Elise. This has been recorded. It will be put onto the Hihi News YouTube channel within the next couple of days. So if you want to rewatch anything, if you want to share it, please do so. So it will be there at least, and probably in many other places very shortly. Thank you everybody for joining. Fantastic audience, fantastic questions. Over to you, Elise.
Karakia whakamutunga, kia hihiri tātou. Ka whakairia te tapu, kia wātia ai te ara, kia tūruki whakataha ai, kia tūruki whakataha ai. Haumie hui e tae ki e. Restrictions are moved aside so the pathway is clear to return to our everyday activities. Kia ora.